Hi class, this is your instructor Dr. Hemangi Patil, and I'm constructing this video to introduce you about plasmid mapping and how to go about it step by step. So let's begin. Before we go into plasmid mapping, let's talk a little bit about DNA itself. As you already know, DNA is a double helical structure, and what we see here are these two strands of DNA, which is deoxyribonucleic acid, and these two strands are actually anti-parallel. That means they have an orientation that we talk about. This will be 3' end, and let's see where it ends. It moves this way, and this will be 5' end. That means this is where your phosphate is going to be, and this is where your hydrox group is going to be. And then this will be 5' end because this is 3'. That's what we called anti-parallel. And this strand is moving this way. So this will be 3'. So this will be your OH end, and this will be your phosphate end. So two independent strands of DNA, and they are connected. The nucleotides with the nitrogen bases are connected using the hydrogen bond. So what we see here, these are hydrogen bonds between the nitrogen bases. And there are 4 nitrogen bases in DNA: adenine, which we say A usually, and then thymine, which we say T, cytosine C, and guanine G. Now, if you recall from your previous lectures, the Chargaff's rule, we know that A will connect to T, adenine will bind to T using 2 hydrogen bonds. Now, these are weak bonds. These bonds do not require sharing of electrons. So these bonds are easily breakable using heat. These are again hydrogen bonds, and you can break them using heat. Similarly, G, which is guanine, will bind to C, which is cytosine, with 3 hydrogen bonds. So again, G and C binding is a little bit stronger than A and T binding. So you will need more heat to break G and C binding as compared to A and T binding. And this is what we see here. So this is adenine right here based on the color, and this is thymine, and there will be 2 hydrogen bonds between them. So again, going into basics, each of this DNA monomer, because it's a polymer, it's made up of the same things again and again. So each of this DNA monomer is a nucleotide. And again, this nucleotide can have adenine, thymine, cytosine, or guanine, depends. So this nucleotide that is boxed right here will have a phosphate group, which will be always at the 5' end, will have a sugar, pentose sugar connected to the phosphate group, and this is the hydroxide group, which will be at the 3' end, and then the sugar will be connected to the nitrogenous base. Now in this picture, the nitrogenous base is adenine, but as I told you, that nitrogenous base can be thymine, can be cytosine, guanine, or adenine. Now that's for DNA, but there is another nitrogenous base that comes into picture when we talk about RNA, and that is uracil. So RNA does not have thymine, it has uracil instead. So in RNA, A will bind to U and G will bind to C. Again, A and U will have 2 hydrogen bonds, and G and C will have 3 hydrogen bonds. So that's the basic structure of DNA. Again, just to review, DNA is double-stranded helical structure. It is a polymer, that means it's made of the same units repeating all over. And these units are, we say A, T, G, C. These are the units. But when we are talking about these units, these monomeric units are actually the nucleotides. So they look like this. There's a phosphate head, pentose sugar, and a nitrogenous base. This backbone that you see right here, this backbone is actually made up of sugar and phosphate. So this sugar and phosphate together, that's what makes the backbone of DNA. And these nitrogenous bases, they hang out and they connect to another, the complementary strand of DNA using hydrogen bond. And AT has 2, GC has 3 hydrogen bonds between them. So now that we know about the DNA strand, again here in this picture, what I'm showing you is a phosphate head and then the sugar, and you can see how the phosphate sugar backbone actually makes the DNA strand right here, while the nitrogenous bases hang out and then they pair up. So there are two hydrogen bonds, 1 and 2, between A and T, and then 3 hydrogen bonds between G and C. So this is just pictorial view of it. Now, if you noticed, A and G are actually wider. They have two ring structures. These are called purine. So A and G are actually purines. And T and C and also U in RNA, these are called pyrimidines, and they have only one ring structure. So if you notice, a purine will bind to pyrimidine, a purine will bind to pyrimidine. Two purines don't bind to each other, two pyrimidines don't bind to each other. And that allows DNA to keep the width between the two strands the same. All right, so we talked a little bit about DNA. Let's talk about restriction, digestion, and how an enzyme cuts it. So this is a sample DNA, and you have these complementary nucleotides binding. The restriction enzyme shown in this picture is HIN3. It's just an example, but this restriction enzyme will recognize a sequence of DNA, and when it most of the time these sequences are palindrome sequence. That means if you read it from this side or this side, they are going to read the same. So these palindrome sequences, when they are read by these restriction enzymes will recognize it and will cut it in a pattern. Now, depending on the restriction enzyme, either it can cut straight, which will make sticky ends because the DNA will be something like this, no hanging sequences, or it can make, these are, sorry, blunt ends, and or it can make sticky ends which look like this. So there will be some nucleotides missing here and here, which is what HIN3 does. If you notice, HIN3 actually makes sticky ends. So these are sticky ends, and that's how restriction enzyme works. Now, why do we need to cut the DNA using restriction enzyme? Well, there are a million reasons why we need to do that, but this slide shows you one of the reasons that we are using in our lab. Okay, and that's cloning basically. So you have a gene of interest, and it could be any gene, say insulin or GFP, that you will be using in your lab, green fluorescent protein from jellyfish. So you have the gene of interest and you have a plasmid of interest. So when you cut the plasmid, it will make these sticky ends using restriction enzymes, and you can see these hangovers right here. And then you use the same restriction enzyme to cut the gene of interest, and you will have these sticky ends with the gene of interest. Now, when you mix these two together, these sticky ends will do the job for you. They are complementary because they are cut using the same restriction enzyme. So they complement each other. So for example, if there is A here, you're going to have T here, so that they complement each other. And then when you mix that, you're going to see this binding. Sometimes when you have blunt ends, which are, no hangovers, of course, with the blunt ends, you can use enzyme ligase to kind of stitch these two pieces together. But if there are sticky ends, that process expedites and you have an easy binding of a gene of interest or to your plasmid. So this is one of the main reason why restriction enzymes are used. So now that you know how the restriction enzyme is going to help make a clone, here are two possibilities, but there are more possibilities, of course, but here are two possibilities that can happen. The gene of interest, right here in red, squiggly line, this is your gene of interest. It has inserted into your plasmid successfully. Now, yes, this is a good plasmid, but what if the gene of interest got inserted twice into the plasmid. So now the plasmid size in the base pair context will be more. So if this is 1,000 base pair and your plasmid is 4,000 base pair, now your plasmid should be 5,000 base pair. But if it is not right, then it will be 6,000 base pair in this case, because there will be 1,000, 1,000, and then 4,000. So it will be 6,000. So based on the size of the plasmid, you can tell if there is one gene of interest inserted or two genes of interest inserted. Also the orientation. What if this was 3' here and 5' here, but you wanted 5' and 3' insertion. So if the insertion is the alignment or the direction of the insertion is not correct, that also you can evaluate by plasmid mapping. And therefore plasmid mapping has a very high importance in cloning, is because you want to verify the product that you get after inserting the gene of interest into plasmid is the correct product. So that evaluation requires you to do the plasmid mapping. So on this slide, let's talk about what happens with the plasmid mapping. Say, for example, we are having, this is your plasmid, and then you're taking an example of EcoR1, which is a restriction enzyme, and EcoR1 cuts this plasmid, which is of 5,000 base pair or 5 kb, we say, cuts this plasmid only once. So if EcoR has only one cutting site on this plasmid, then after the digestion is over, it's going to make this plasmid a linear strand of DNA, which is of 5,000 base pair size. So what if another enzyme, say BAMH1, has 3 cutting sites? So it's going to cut here, let's cut here, and then here. And then if it is cutting, say 1,500, 500, and then 3,000. That will be the cutting sizes, just as an example, because they need to add up to 5,000. So 3,000 plus 1,000, that's 4,000, then one more 1,000, that's 5,000. So the size will not change, the total size will not change. But if BAMH1 is cutting 3 times, now you're going to get one strand, which is 3,000 base pair, then another strand, which is 1,500 base pair, and then a small strand, which is a 5,000 base pair. So you're going to get 3 different DNA strands based on it. Now, this is just with one enzyme cutting. So with one enzyme cutting, you can find out the size of the plasmid. You can verify that, and that will tell you how many genes of interest have been inserted. That's a good idea, right? And then for the orientation purpose, to verify the orientation of the gene of interest, you cannot use just one enzyme. So you will have to use 2 enzymes. So what you can do is you can have a plasmid and you can cut it with EcoR1. And then say you're cutting with BAMH1. So BAMH1 is cutting here, here, and here. This is your 500. This is your 3,000 with BAMH1. And if you notice this 1,500 fragment that which was getting cut with BAMH1, now it is getting also cut by EcoR1. So let me go ahead and write BAMH1 here so that there's no confusion. And then now EcoR1 is cutting inside the 1,500 base pair fragment. So say it's cutting what's this, 700 here and 800 here. So now with the double digest, we call it double digest because it's 2 enzymes cutting, we are going to have the fragments 3,000. We're going to have 500, we're going to have 800, and we're going to have 700. Now, if you take a look at it and you have single digest from EcoR1, we have a single digest from BAM H1, and then we have a double digest from EcoR1 and BAM H1 together. Now, if you take a look at that, you will see that here you have 3000 and here you have 3000. So you can say, oh, this is the same segment. And then you have 500 and 500, and you can see, oh, this is the same segment. But then you have these two, which add up to the missing piece from BAM H1 restriction, which is 1500. And with that, you can say, oh, that means the 1500 segment is where EcoR1 must be cutting. And it must be cutting in such a way that it's distributing this into 700 and 800. And that's how you kind of fit the puzzle pieces together. Now, on the next slide, we will take an example, the actual example of the restriction digest and for plasmid mapping. So let's move to the next one. So on this slide, we have an example of various different band sizes given for each of the restriction enzyme. If you notice, EcoR1 has three segments, HIN3 has one segment, and when you have a double digest with EcoR1 and HIN3, you have four segments. So always go for the less number of segments and lay it out. So I'm going to say here, this is where your HIN3 is cutting. So say HIN3, and because it gives you only one band, that band actually tells you the size of the plasmid, which is 50 kb in this case. Now I'm also going to check that 50 is the addition of fragments from HIN3 and also addition of the fragments from EcoR1. That means there is no fragment in the EcoR1 that is double, because sometimes you may have double fragments and then your size will not add up to 50. And that's an indication that you may have a double fragment or of the DNA. And when you add up the double digest, that also gives you 50. That means there's no double fragment here as well. So we are done with HIN3 mapping. Let's map EcoR1 next. So for EcoR1, so EcoR1 is cutting here. And then it has 30, that's the first fragment. So I'm going to say half of 50 is 25. So if I make a cut here, that's 25. So 30 must be a little bit more. Again, it's just an estimation. So this is 30 base kb right here. And then the second fragment is 15. So 15, that's about this size, 15 kb, again estimation, and then 5 kb. So these are the three fragments you're getting with the EcoR1 digest. Now, when we do the double digest, we always take the highest number of fragments and lay them out. So you go ahead and lay out the EcoR1 digest. And write EcoR1, that's where it's cutting. EcoR1, that's where it's cutting. EcoR1, that's where it's cutting. This fragment is 30, this is 5, and this is 15. So I laid out the EcoR1 fragments. Now I am going to overlap HIN3 fragments on top of it. So when I look at the double digest and I see there is 15 here and 15 here, that stays intact. That means HIN3 is not cutting inside that 15 kb fragment. There's 5 here and 5 here. That means that fragment also stays intact. But there's this 30 fragment which is actually getting split into 20 and 10. So your HIN3 here is actually cutting between that 30 fragments. So I'm going to go back and say this is my 30 fragment, and then you have split into 20 and 10, and I'm going to cross out 30 because it got cut, and this cut is with and 3. And that's how you do the plasmid mapping for double digest. Now, please understand that I'm putting 20 on this side and 10 on this side, but that is an estimation. We do not know for sure if 10 is on this side or or the other side, and for that we will have to cut it using a third restriction enzyme to be very sure where exactly 20 and 10 fragment is. It could be just reverse. So this is how you do the double digest. If you submit this, again, there is yes doubt where 10 is going to be, where 20 is going to be. Their orientation is not certain, but it does support the bands that you see for single digest and double digest, so it is a good estimate.